Our next speaker is Dr. Chris BOWLER, Research Director at the CNRS and Director of the Plant and Algae Genomic Laboratory at the Institut de Biologie de l'École Normale Supérieure de Paris. Désolée, j'ai switché en anglais. Of Paris. So, Chris BOWLER is someone that you will necessarily meet when you work in plant biology. He has been starting his career on plants, and with time included also phytoplankton. So he received a PhD from in Belgium, then did a postdoc in the US, and got a permanent position in Naples, in Italy, and arrived in Paris 20 years ago. He has been member of the EMBO, received the CNRS Silver Medal, 2 ERC Advance Award, Grand Prix scientifique de la Fondation Louis D de l'Institut de France. He was a fellow at the Radcliffe Institute and also Radcliffe Institute of Harvard University. He was elected member of the French Academy of Agriculture, and he held the chair of professor in biodiversity at the Collège de France. So very little responsibilities, right? So regarding research, his main interest is in understanding the responses of plants and the diatom to environmental signal through genomics. And since 2021, he's the scientific director of the Tara Oceans Project to explore the biodiversity in the oceans. So please.
Merci, thank you. Thank you for the introduction. We'll wait for the slides to switch to my slides, yeah. And thank you for being here and attending this conference. It's a very, very nice conference. You're very, in fact, privileged to have such a conference to listen to, and that explores many different aspects of environmental sciences and climate and big important topics for today's society. So thank you to the organizers, and thank you also to the participants for joining here. So I'm going to talk about the oceans today. I'm maybe the only person at this symposium talking specifically about the oceans. I call the ocean the multiverse because it's really multi-dimensional, as I'll try to show you in different ways, and is also an environment where the physics, the chemistry, the biology really interact intimately with each other, and is a beautiful system to study. Even though complex, it's maybe less complex than things like soil, which I would never touch personally due to its complexity. So I'm going to talk principally about the Tara Oceans project and what we've learned from it. But first of all, let's begin with a little introduction to the ocean. And you're all familiar with images like this coming from space. Even though we call the planet Earth, it is mostly blue due to what we see as the water on our planet. But in fact, water is relatively scarce. If you take all of the water out and put that superimpose it onto a dry Earth, there's actually not that much water. This represents the ocean, the water in the ocean, in this bubble here, and even smaller are the cryosphere and the fresh water, which Bastien will talk about after me. So these are very precious resources and relatively scarce. However, we now appreciate that life appeared in and is sustained by the ocean in many, many different ways. The microbes in the ocean perform half of the photosynthesis on our planet, remarkably. The ocean captures a quarter of our CO2 emissions, captures 90% of the heat generated from greenhouse gases. And this is how the ocean regulates the temperature on our Earth, and yet it is still pretty poorly known. So our contribution to trying to understand something more about the ocean was through our work with the Tara Ocean Foundation, Fondation Tara Océan, which is a French organization, funded to some extent by Agnès B., the fashion designer, but not only. And they have a research sailboat called Tara, which you see here, 36 meters in size, which they give to scientists like us to go out and do what they consider to be important scientific projects for better understanding of the ocean. So I got involved with the foundation now more than 15 years ago, being involved in a 4-year expedition called Tara Oceans from 2009 to 2013, where we did a global sampling expedition to explore marine planktonic ecosystems. We have a lot of non-scientific aspects of the project related to popularization, communication of science, education, improvement of ocean governance through interactions with politicians, policymakers, and so on, which I'll not go into. But I will really talk specifically about the scientific aspects of the project. So, first of all, a little introduction to this world of marine planktonic ecosystems. What are we talking about here? So, if you think about life in the ocean, you may struggle to wonder, what actually is life in the ocean? If you go for a walk in the forest, you can see the biodiversity around you. You can see the plants, you can see the forests, you can see the animals and the birds, and we can appreciate that very, very easily. But in the ocean, we see dolphins and whales and corals and stuff like that on the TV, but that is actually not where the biodiversity is in the ocean. And just as Marc-André showed us for the soil, also in the ocean, perhaps even more so in the ocean, the biodiversity is in the microscopic world. When you take a microscope and you look at a drop of seawater, you see this explosion of life in the ocean, which is absolutely amazing to look at. There's all kinds of shapes and sizes and different kinds of organisms that you can see here. You just need a microscope to explore it. So these organisms are very small. They will all fit on top of a pinhead. They begin with organisms a couple of millimeters or so in size, and they go all the way down to the bottom here, covering the bacteria, the viruses, which are even smaller than you can see here. They're very small, however, they represent the whole tree of life that we know of that exists on our planet today. In particular, beyond the viruses and the bacteria, when we're talking about eukaryotes, in particular single-celled eukaryotes, which represent single-celled life forms of cells complex like our own cells, there is a whole diversity of life that is present here, which we don't always appreciate. So here we have the eukaryotic tree of life, and if we start down here, this is where we are. We are in the animals down here. And our next closest neighbor in this tree are the sponges. Okay, so maybe if you look at your neighbor, you may think, yeah, that's about right. But this is the evolutionary distance between animals and sponges, just shown here. Then we have the plants, the algae, green algae sitting in this branch here. Then you have all this amazing diversity that is present here of organisms that we really know very little about, okay? And all of these kinds of organisms are present in a drop of seawater. So we have the whole tree of life present in a drop of seawater that we can go and explore. And this is one of the primary interests of what we are trying to do in this project. So we can appreciate that these forms of life are highly complex and evolutionarily very, very diverse. They also represent the majority of biomass in the ocean. If you take a giant net and scoop up all the life in the ocean, we realize that these microscopic organisms represent about 2/3 of the biomass in the ocean, and they perform all kinds of important functions, being at the basis of the oceanic food web. So simply, if there's no plankton, there are no fish, there are no larger organisms. The photosynthetic guys take carbon dioxide, they take carbon dioxide out of the atmosphere and sequester it into the ocean for various periods of time. Through photosynthesis, they also generate oxygen. And because the ocean is generally deep, when these organisms die, a fraction of them will sink to the bottom of the ocean and ultimately generate the gas and the petroleum fossil fuels that we are now burning so actively in our society. So this is one way that the plankton impact climate, by taking carbon dioxide out of the atmosphere and storing it in the Earth's interior for geological periods of time. And also, being biological forms of life, they're also sensitive to changes in climate. So this is one aspect that we also need to learn more about. So this is the basis for studying what we wanted to do during this project. We realized that to do this, we needed to do a sort of systems approach to study the whole community of organisms present in these communities, aiming to understand who is there, what do they do, with whom do they do it, and why do they do what they do. Why is it important for them? Why is it important for the ecosystem? Why is it important for the functioning of the planet? So we did this by working to sample the entire community of organisms, from viruses, the smallest viruses, all the way up to the smallest metazoans of a few millimeters in size. This is spanning 5 or 6 orders of magnitude, the equivalent of sampling in a forest everything from an ant to a brontosaurus. Okay, so you need several nets to be able to catch all of those different kinds of organisms. So that was one important aspect that we needed to establish. We wanted to do this at global level because everything is connected in the ocean. And in order to make sense of our data, we had to do all of this in a very standardized way, doing essentially always the same thing everywhere we sampled. And then using very standardized analytical pipelines to make sense of the data. So that was largely based on microscopy and based on DNA genomics analyses to disentangle these complex, invisible communities that we wanted to look at. So the project ended, the expeditionary part, the wet part of the project, let's say, was completed in 2013. And we were able to publish our first data from the project, reporting some of the first results. Already in 2015, in fact, we were given a special issue of Science by the editors at Science, where they gave us the front cover, they gave us the editorial, and they gave us 5 papers that were published back to back, reporting the first results from the project. So for a scientist, this is a fantastic achievement, as I'm sure you can appreciate. So since then, we've been producing more and more data and doing more and more analyses, and so there's a whole world of things that can be explored and discovered from this data set. So let's just begin with some really, really simple stuff, the sort of things that a 10-year-old child would ask, or maybe an alien coming from outer space would ask us about, who do we share our planet with? For example, which group of plankton is the most abundant? We should know that answer, right? Or which group of plankton is the most diverse? These are really simple questions, which, and you will see the answers were surprising. So let's first, since not many people study the ocean here, let's first make the analogy with those questions answered for terrestrial ecosystems. Okay, so in terms of biomass, in terms of abundance of organisms on land, who dominates? Well, it's the plants. In terms of gigatons of carbon present, the plants really dominate everything else with whom they share the planet. We, within the animals, are really pretty insignificant in this little corner of the plot. So even though we are incredibly powerful, we're not so important in terms of biomass. So it's really the plants that dominate terrestrial ecosystems in terms of biomass. In terms of diversity, the answer is different. In terms of diversity, here we have an image where the size of these different organisms represents their diversity. And the big guy here are the insects. So in terms of diversity, it's the insects that dominate terrestrial ecosystems, okay? So the question that I'm asking really is, who are the equivalent of the plants and who are the equivalent of the insects in the ocean? Okay. And in both cases, the answer surprised us. So in terms of abundance, it's these guys called radiolarians, which turn out to dominate the biomass in the ocean. Radiolarians, we've known about them for quite some time. These are some of the early drawings of Ernst Haeckel that you can see here from the 1890s or so. But we never really appreciated their abundance because they're very fragile organisms. They tend to explode when you sample them and so on. But by doing in situ microscopy in the ocean, we could appreciate the extensive biomass of these organisms in the ocean. So the radiolarians are the most dominant in terms of abundance, biomass in the ocean, which I don't know if anybody guessed that answer. Probably not many of you. It was a surprise to us as well. Even more surprising in terms of diversity, the winners are a group of organisms called diplonemids. Anybody heard of diplonemids? We had not heard of them either. So yeah, we were also surprised to learn that these guys were the most diverse eukaryotic group of organisms in the ocean. We don't really know what diplonemids do because we don't have representatives in the lab, but they're sitting down in this group of organisms in the eukaryotic tree of life. They're close to trypanosomes, leishmanias, so we assume that there's some sort of parasites against other groups of plankton, okay? But we don't really have much information about them. So the radiolarians are the plants of the ocean, and the diplonemids are the insects of the ocean. So those results were already quite surprising to us when, even at this really basic level, we're getting surprising results. Now we can dig a little bit deeper. Because we did this global survey of multiple hundreds of sampling sites, generating thousands of samples, and characterizing viruses and bacteria, eukaryotes, metazoans, all kinds of things, at each place, we can throw all of that data together, and we can do a giant compute and ask, well, which organisms do you always find together? Or which organisms do you never find together? Okay, so using, largely a statistical approach, we can define the social network of this microscopic world, the Facebook of the ocean. And this is how it looks. It's not very pretty. But basically what we have here, every dot is a different organism. Okay, so the blue ones are eukaryotes, the pink ones are bacteria, and so on. And if you have a green line between them, then these organisms co-occur. We find them pretty much ubiquitously together in the same samples. If you have red lines between them, you don't perhaps see them too well. Red lines, which are over here, red lines between them indicates that these are organisms that you never find together. Okay, so We can basically define sort of social plankton. These green guys here, there are some incredibly popular forms of plankton that socialize a lot. These are the I don't know, the Beyoncés, the Mbappés, the Benzemas of the planktonic world. They're super popular with everybody. And then we have these antisocial guys, which are more the Trumps, the Putins, the whatever. Okay, so we can see these things at the macro ecological level. We can tease that apart and look taxonomically. And look at, how many positive associations do we have in this network and how many negative associations do we have in this network. And we basically see that the number of positive associations is much larger than the number of negative associations, implying perhaps that cooperation is more important than competition in this in these communities. This was also a surprise to us because the sort of neo-Darwinian ideas are survival of the fittest, competition, the fight for survival, all of these concepts which are ingrained in our teaching of biology, of evolution these days. But, this sort of data perhaps indicates that, no, that's not how it works, but it's more about cooperation between organisms, everybody working together to ensure that the whole ecosystem works and everybody benefits. So that was a nice message that came out of this planktonic world that can also be a benefit for our own societies, of course, to think in this way. So that's the sort of thing that you can see at this very large-scale macroscopic level, but you can also dig deep into this interactome and look for your favorite organism. Each of these dots is a different organism. So if you have a favorite organism, you can go and look in here and say, okay, it's here. And you can see who does it interact with, what does it do in the world. So this is a beautiful playground to go and discover completely new interactions. One example I'm going to give you about that concerns this dinoflagellate photosynthetic organism called a Symbiodinium, which we know is a very famous symbiont in all kinds of symbiotic interactions in the ocean. It's the Symbiodinium dinoflagellate that gives the beautiful color to coral reefs and forms all kinds of symbioses with all kinds of marine mollusks, giving them all kinds of amazing colors. But the question that we could ask here is, well, does Symbiodinium also form symbioses with organisms in the planktonic, in the floating world of the ocean, not just the fixed benthic world? And again, we had a surprise that yes, it does. Symbiodinium, in fact, has found a house to live in, and that house is another organism. The other organism is a ciliate, a planktonic ciliate. You can see it here in the microscope with all of its hairs on the outside. It's a single-celled organism, about 100 microns inside. And if we look inside these guys with the microscope, we can see they're completely loaded with Symbiodinium cells, which are indicated here in red and dark blue. So Symbiodinium has found another house to live in while it's floating around in the ocean. We can pull out these cells and we can sequence them and characterize them genotypically, and we can see different genotypes, and we can see that they're pretty much present ubiquitously around the ocean and present in different genotypes, and presumably this is how Symbiodinium travels around the world in its little house, its little car. And when it finds a nice coral reef to go and inhabit, it will jump out and go and infect the corals instead. So that's another beautiful example of the sorts of things that we can discover by playing around in this microscopic world. Over the years, as a consortium of scientists, we have published a lot of papers together exploring this data set. Oftentimes we get the front cover of the journals, which is always satisfying. And even more satisfying was a recent editorial published in the journal Nature on 8th of June of last year, where they talk about the Tara's inspiring 20 years at sea, really talking about the fantastic discoveries which we have made, and that is, of course, a great achievement as a scientist to get that level of recognition for all of this often very difficult, very challenging work that we've been performing over the years. So one aspect that I, of course, need to talk about here in this symposium is what can we learn about climate change, and that is what I'll tell you about in the remaining few minutes of our talk here. So, of course, we know now how powerful we are. We are a biogeochemical force on our planet, equivalent to hundreds of volcanoes going off at the same time. Even though in terms of biomass we are not that important, but we really can consider ourselves as a superpower. And this is very much also manifested in the ocean as well. Our oceans are sick. We can see this through actual data. Some of this generated from Copernicus, from the remote sensing data that you heard about yesterday from Hervé Jean-Jean. And so, for example, we have absolute data showing how sea surface temperatures have increased since 1900, how sea level rise has increased, has gone up since 1900. The summer sea ice has lost about half of its surface over the last 50 years or so. And seawater is slowly acidifying from a simple chemical equation of CO2 plus H2O makes carbonate and bicarbonate, which are acidic. We have actual data, because we have probes in the ocean that measure acidification since the 1990s. And we can see from this actual data, how acidification is slowly increasing, both in the Pacific and in the Atlantic Ocean. So all of this is real data. It's not models, it's not simulations, it's not political blah, blah, but it's actual scientific data. What is even more disturbing is what happened last year, which was also shown yesterday in a couple of slides. And this, again, from the Copernicus data, beautiful data set, beautiful resource for us scientists. We can see how sea surface temperature of the global ocean, this is 60 degrees south to 60 degrees north, essentially all of the global ocean. We see how through a yearly cycle that sea surface temperatures have steadily increased from the 1970s up to the 21st century. And last year, something happened. This is up to end of November, 1st of November 2023. And we see that sea surface temperatures have hit new records, and we seem to have had a step change, and we don't really completely understand what is going on here. So there's a lot of things that are very concerning about the ocean that we can see from actual data. But we need to try and understand that. We need to try to understand what is going to happen to ocean life as a consequence of climate change. And I'm going to give you two examples of the sorts of things that we can do. One example is digging into the past to get evidence for the effects of ocean acidification on life in the ocean. So this is a bit of a challenge, because what we would ideally like to do is to compare the ocean today with how the ocean was 200 years ago, okay, before we started injecting all of this CO2 into the atmosphere. Unfortunately, we don't have samples of the ocean from 200 years ago, right? We don't have a time machine to go back and get those samples. So it's pretty difficult to find such samples, but we do have one example of a set of samples that we have access to today, which comes from the very first oceanographic expedition from the 1870s on the HMS Challenger. They set out, did a 5-year expedition similar to ours, looking at ocean life, and they collected a lot of samples and brought them back, and fortunately they were archived in the Natural History Museum in London, and we have access to them to compare those samples collected 150 years ago with the samples collected on Tara. And in this study, we're particularly interested to look for the effects of acidification by looking at the process of calcification in foraminifera organisms. These are single-celled eukaryotes again, which have calcified cell walls, calcaire, and we know that this process is sensitive to pH. So in this study, basically using advanced microscopy techniques, the authors compared the thickness of the cell walls of the foraminifera collected on HMS Challenger with the foraminifera collected by Tara 150 years later. And in the blue here, you can see very clearly how the thickness of the calcified shells has decreased over this 150 years of time since these two samples were collected at the same time, at the same place in the Pacific Ocean. This study reports a quantified study of these processes. And this is one of the very, very few evidence that we have of how acidification is really impacting ocean life. There are really very few examples like this where we see direct effects of what is actually going on. So I think that's an important example that I wanted to share with you. And then the other sorts of thing we can do, in addition to going into the past, is to look into the future, to use the data set from today to see how that may change as we go into the future. So, and here is a study where we projected changes in plankton diversity by end of 21st century, essentially from the global sampling that we performed on Tara Oceans. We were able to generate global maps of the diversity of different kinds of organisms, for example, photosynthetic bacteria, for example, parasitic protists, and so on. And we see these color codes by diversity, and we see the general observation of diversity being highest in the equatorial regions and tapering off as you go towards the higher latitudes in the Arctic and the Antarctic. This is classic latitudinal diversity gradients, first observed by Alexander von Humboldt more than 200 years ago in terrestrial ecosystems, now seen for the first time in the ocean. So we have these maps of how plankton biodiversity looks in today's ocean at the beginning of the 21st century. We can take this data, this information, and we can inject it into the ocean that is predicted to occur by end of 21st century from the IPCC models that Valérie Delmotte introduced you to yesterday. We can take these projections of how sea surface temperatures are predicted to change by end of century, how photosynthetic activity is projected to change by end 21st century. We can throw today's plankton into this simulated ocean and see how they separate out. And here we have basically plots where I'm showing you projected diversity anomalies of these different plankton groups by end of 21st century. And if you just take this example of copepods, a group of zooplankton, we can see how diversity is projected to decrease at lower latitudes and to increase at higher latitudes. And you can see basically that there's a pretty dynamic changes appear likely to occur by end of 21st century. As Marc-André Sellos told us this morning, it's not just that high biodiversity is a great thing and low biodiversity is a bad thing, but biodiversity needs to be in equilibrium with the environment. And any change in biodiversity is potentially a worrying thing. So here we see these anomalies both in a positive and in a negative sense. And if we map that out by equatorial, by latitudinal bands, where the projected anomalies are most likely to occur, they are at the higher latitudes in the north and the higher latitudes in the south. And if we compare these projected anomalies with key processes in the ocean, such as carbon export, so the capacity of the ocean to suck CO2 out of the atmosphere and store it at depth. These are very important areas for carbon export activities. These are very important areas for fisheries, which are dependent on plankton, of course. And these are also areas where we have a lot of marine protected areas. These are areas where we try to protect the biodiversity in the ocean. And again, these projected anomalies that we predict may occur by end 21st century need to be taken into consideration in our planning of, where do we put the marine protected areas to try to conserve that biodiversity as we move into the future. So that's more or less what I wanted to talk to you about in summary. Basically, using this systems biology approach, I think we've been able to shed light on this complex, invisible ecosystem at global scale and get some indications of how this works. There are multiple new discoveries that continue to be made, even completely new phyla of viruses continue to be discovered by exploration of this data set. And because the resources are open access, many people beyond us are making those discoveries. And as I showed you, because we have this global data set from today's ocean, this is sort of a baseline from which we can try to better understand what the effects of climate change will be on marine ecosystems in the future. So I will end there and take any questions and thank our sponsors for supporting the expeditionary part of our work, the agencies who have funded the actual research that we've been performing, and the group of different organizations, scientific organizations that have been involved in exploring this amazing data set. So thank you to them and thank you to you for your attention. I'm a little over time, but...
Oh, thank you. Questions?
Hi again. Thank you very much for your presentation.
You can watch these videos. If you don't want to listen to the questions, then you can just watch these. But yeah, go ahead, please.
So you talked a lot about the climate change effect, but I was wondering, how about the plastic and the microplastics effects on the biodiversity?
Yeah, thank you for the question. Yeah, the ocean is full of microplastics and nanoplastics. Is getting worse and worse. They're also present in the Arctic, in the Antarctic, because everything is connected. So there's a lot of plastic material out there, slowly decomposing. It's obviously bad news. There's a lot of toxic impacts of those plastic particles, fragments in the ocean. But it's also an opportunity to see evolution in action, because the presence of these microplastics has also stimulated the evolution of mechanisms in bacteria, in particular, to use the plastic as food. It's full of carbon, right? So the microbes, again, these amazing microbes, they have evolved. They're evolving ways to utilize that carbon to digest it. It's a very slow process, it's very inefficient, but we are seeing mechanisms that have evolved in the microbial world to digest the plastic, and that gives us, new possibilities to perhaps use those genes, use those enzymes, and maybe devise new methods to tackle this huge, disastrous impact of plastics on our planet and on our ocean. So yeah, it's bad news, but not all bad news. Yeah. Okay. There you go. Some plastic.
Thank you, Chris, for this very nice talk. I have a question about actually the different scenarios about the increase in temperature. So you showed some results on what the impact on biodiversity on plankton, but what is really the difference we could see with the different scenario? Let's say, I guess this is the normal, the average scenario, but what will be the expected impact with, let's say, the highest, the most terrifying scenario, for instance? Yeah.
Yeah, these were actually not the most terrifying scenarios. They were done a couple of years ago before the more terrifying scenarios came out. So they're fairly conservative studies that were performed here. There's a lot of details that I didn't go into, of course, from looking at this global scale view of the projected changes. And this study, of course, did not take into consideration also the evolvability of the organisms, their capacity to evolve, to adapt to new conditions. And some of the organisms are going to find their way quite easily by evolving new mechanisms, new adaptations, new acclimations to new conditions. Some will be able to migrate to find, their sweet spot of nutrients and temperature and oxygen and so on. And others, the more exotic guys, will disappear. As we see, as Brazilian rainforest is converted into soybean fields, we see sort of similar things going on. So there will be a lot of changes in the ocean coming. Life will go on, of course. Life wants to live. So life will go on in the ocean without a problem. The problem is, the ecosystem services that the ocean provides, in particular for our societies, how that's going to impact us. And there will be a capacity for adaptations, but what is perhaps the most disturbing in this story is what will happen in Arctic and Antarctic, because these guys have nowhere to go. The other organisms perhaps can migrate, but the Arctic, the Antarctic are end games, basically, end of the road. And we see from looking at not only the community of organisms and the structure of those communities and the genes that are present in those organisms, that Arctic and Antarctic are really different worlds. It's not just a gradient from tropics to the poles of life, but there's really different ecosystems, really different organisms, different mechanisms that underpin the biology of those ecosystems that we risk losing. So I think there is an imperative to study those ecosystems, in particular to study life in the sea ice, which essentially nourishes the whole ecosystem in these polar regions. To study that, learn more about how it works, to eventually see, how we can try and protect it in different ways. So yeah, it's of course life will go on, but it will be a very different ocean, impacted also by changing currents, changing circulation, changing oxygen levels, many things. So.
Yes, yeah. I have a question. Thank you very much for this very interesting presentation. But you present some data corresponding to plankton at which surface? Because I'm wondering if the impact of climate change is important on the surface of the ocean, of course, but what happens deeper?
Yeah, very good question. Due to the small size of our research vessel, we could only sample the top 1,000 meters of the water column. Okay, so to go deeper, you need bigger ships, real oceanographic ships that have the capacity, that have the cable to send things down much deeper. So we only did the top 1,000 meters. The light penetrates in general about 100 meters only, so we studied the photic zone pretty well. and then the zone immediately below that pretty well. But the average depth of the ocean is 3.7 kilometers. That's the average. The maximum is like 12,000 meters. So there's a lot of ocean there which we did not study. And there's a lot of ocean that is very, very important that we know much less about, in particular the deep ocean ecosystems, which are out of sight, and so typically we think they're not so important, but in fact they are extremely important. They're typically the end of the road for everything that sinks to the bottom. And the more we modify the surface ocean, the more that will impact the deep ocean, of course. And also, if we're thinking about carbon dioxide removal strategies by increasing biomass in the ocean, or growing seaweeds, or doing agriculture in the ocean, or all kinds of strategies being proposed there, all those different things are likely to impact the deep ocean. And what goes down eventually comes up. It may take thousands of years, but the fact that everything is connected in the ocean, not only horizontally but vertically, means that we need to think hard about the three-dimensional component of the ecosystem there. Yeah.
Any other questions?
Well, thank you, Chris, for your presentation, and we will